Greetings. My name is Don Cowan. I'm the Director for the Centre of Microbial Ecology and Genomics at the University of Pretoria. And right now I want to take you to Antarctica. Antarctica, that's that huge continent at the bottom of the world, almost completely covered in a huge ice cap. But the parts I'm interested in are the ice-free areas. And these occur on the peninsula and around the periphery, around the coastal areas of the continent. In most of Antarctica, particularly the McMurdo Dry Valley area where I work, this is a very extreme environment. It's so extreme that the only life that exists there is microbial. There are a few lichens, there are a few mosses. The nearest higher plant is 5,000 kilometers away. These organisms play a very important role. In the absence of higher plants, the microorganisms are doing everything in the soil. It's called ecosystem servicing. They're fixing nitrogen, they're fixing carbon, they're metabolizing, they're performing the services within that soil, and it's solely microbial. So the story of microbiology, particularly in the dry valleys, which was one of the first areas ever worked, this was performed principally by American scientists as part of the early studies pre the Mars Viking missions. They were using the dry valleys in Antarctica as a model system for what they might find when they got the Viking lander onto Mars. And it wasn't until around about 2000, the turn of the century, that modern molecular phylogenetics was introduced to a study of microbial ecology in Antarctica and many other places. And this is about the point that I entered the field of working in Antarctica as well. And so I was able to bring some of these modern molecular methods to study the true diversity of organisms within that environment. And we've done much more to go. And we found some really exciting things. We found, for example, that the diversity was very, very much greater than ever thought before. Culture-dependent studies only give you a fraction of what actually. We've looked, for example, at the diversity at the function of organisms, the extent to which they undertake those processes I mentioned, carbon turnover, nitrogen turnover. We've studied things such as the resilience of these communities to climate change. This is very important. Antarctica is warming as fast as anywhere else in the world, and that's going to change the environment and therefore the microbiomes. And there's a very, very strong conservation remit within Antarctica. This is the last pristine continent. There are no permanent residents there. We currently have a very big an international collaborative project with Australian researchers and others looking at the biogeography of microorganisms across the entire continent. Biogeography means understanding the diversity across spatial scales. And so we are getting samples from researchers all around the world from all areas, and we're going to compare the microbial diversity and try and understand how the microbiomes of the soils around Antarctica differ depending on region, so that we can understand how conservation should be applied. And the last and very exciting thing that we're doing is understanding or working with researchers, particularly at Monash University, to understand how these organisms in very extreme environments around the periphery of Antarctica live in the absence of phototrophy. In other words, they're not using sunlight like plants. And there's a new system which has been starting to be understood, which is called trace gas chemotrophy, where they're actually using hydrogen and carbon monoxide to drive energy acquisition, CO2 acquisition. And that's a very exciting new development in the field of extreme microbial ecology. So watch this space.